about 50 to 70 doubling. After that, the cell lost the capacity to divide, and after that they enter the senescence phase. The senescence has been ascribed to progressive shortening of telomeres at the end of each chromosomes. So by each doubling we have shortening of this area, and this is the phase of after about 70 doubling we have senescence phase of the normal cells. Short telomeres is recognized by the DNA repair system as double-strand DNA breaks. Okay, our DNA repair system recognizes this area as a break and the DNA damage. And finally, it can cause cell cycle arrest and senescence, and this pathways is an affection of P53 and RB genes. So the normal P53 and RB genes are required for the senescence in the normal cells. But in cells in which P53 or RB genes are mutated, we have another pathway that we call it non-homologous end joining pathway. Okay. This is very important pathway, and this pathway is activated in the mutated situation of P53 and RB genes. In this pathway, we have joining the shortened end of two chromosomes. This inappropriately activated repair system result in formation of some chromosomes that are decentric because of this fusion. These are decentric. And after that, they are pulled apart in the phase in the anaphase stage of mitosis and meiosis, resulting in new double-strand DNA break. The resulting genomic instability from the repeated this pathway is called bridge fusion breakage cycle. So this fusion and the breakage, we call this cycle bridge fusion repeated breakage, bridge fusion breakage cycle. Eventually product this by apoptosis in normal situation. But in the cells that have disabled checkpoint, which are mutated, DNA repair pathway are inappropriately activated by their shortened telomeres, leading to massive chromosomal instability and mitotic crisis. The tumoral cells can reactivate the telomerase, so they can achieve immortality because of this enzyme. And also some other tumoral cells have some alternative mechanism for telomere elongation. So we need telomere elongation in the tumoral cells. This tumoral elongation can be because of telomerase enzymes in most of tumors. And in the few tumors, we have other pathway for telomere elongation. Okay. We know that in about 85% to 95% of cancers, this is due to upregulation of telomerase, and in about 5 to 10% we have other alternative pathway for elongation of telomere area. Telomere maintenance is seen in virtually all types of cancer. As I said before, in 85 to 95% is because of upregulation of the telomerase enzyme. And briefly, we can say that in immortalized cancer cell, telomerase is usually reactivated and telomere length is stabilized and allows cells to proliferate indefinitely without any limiting. Okay, and now this is very important picture, and in this area you can see the normal cells and senescent cells, for example here, This is a normal somatic cell. As I said before, in the normal somatic tissue, we have inactive telomeres. So after multiple cell division, about 50 to 70 division, we have shortened telomere. This red area are telomere, okay? I said that they are present in the both ends of chromosomes. So this area are telomere. Now I want you to read this slide, and after that I chose one of you for explaining this slide, okay? Because this slide, this pathway is very important, and all of the points that I said before is summarized in this picture, okay? Done? Are you ready? Are you ready? Yes.
So over here, like you mentioned,
Can you come here? Okay. The other important feature of the neoplastic cells and the other hallmark of the neoplastic cells is development of sustained angiogenesis or angiogenesis, okay? We know that even with all the growth advantage and all the features of tumoral cells, limitless proliferation, evasion of cell death, and all of this, tumor cannot enlarge more than 1 to 2 millimeter unless they are vascularized. So without vascularization and without no angiogenesis, the tumoral cells can enlarge about 1 to 2 millimeters. So, and this is like normal tissue. You know that the tumoral cells require oxygen, nutrients, and removal of waste product from this area, so they need vessels. They need neoangiogenesis.
Yeah, activation of PDGF and IGF, right? Activation of IGF and PDGF.
Growth factors, you mean? Yes, we have some growth factors that one of the important of them is VEGF for producing the new vessels from the other vessels. The cancerous cells, malignant tumor, and also large benign tumors can stimulate neoangiogenesis, and the new vessels sprout from previously existing capillary in that area. We know that the tumor vasculature is abnormal. The vessels, the new vessels are so leaky, are dilated, and their pattern of connection is very irregular and haphazard. But angiogenesis is required not only for continuing the tumor growth, but also the tumor needs vessels for metastasis. The malignant tumor needs, because we said that the most important feature that differentiate the malignant from benign in the neoplasia is ability to metastasis, okay? We know that neovascularization has a dual effect on tumor growth. Perfusion supply the needed nutrient and oxygen, and also newly formed endothelial cells can stimulate growth factor of adjacent tumor cells by secreting many types of growth factors like IGF, PDGF, and also VEGF. Angiogenesis is a necessary biologic correlate of neoplasia both in malignant and benign. Early in the growth of the malignant cells, most human tumors don't induce the angiogenesis. And before the angiogenesis, they remain small or in situ for years, sometimes for years. They are in situ until the angiogenic switch terminates this stage of quiescence. For example, about the epithelial tumors, squamous cell carcinoma, for example. For squamous cell carcinoma, at first we have a normal squamous area, okay? The squamous epithelium is like this, you know? We have some basal cells, like this. And after that, we have maturation of these cells to the mature and differentiated squamous cells. By maturation, we have smaller nuclei with enlarged and more abundant cytoplasm, okay? This is a normal area and normal maturation of an squamous epithelium, like this. And sometimes we have mitosis only in the basal cells. Okay, as we said before, in the normal situation, we have sometimes loss of these superficial cells. For example, in our skin, we have a loss of many keratinocytes, many squamous cells in every day. Okay, and for replacement of these cells, we have proliferation of some cells here. That some of them are the stem cells, or some cells like the stem cells. So sometimes we have mitotic figure like this, only in the basal cells. This is a normal situation. After some mutations, as we said before, the heart of neoplasia and malignancies mutation, and also some environmental agents that are carcinogens. So after this, we have mutation in, for example, these cells, and these cells start to proliferate here with many mitotic figure, for example, like this. For example, all of these are mitotic figure, and we have enlarged polymorphed nuclei, bizarre-shaped nuclei here, for example. Sometimes we have multinucleation. Again, for example, mitosis here, we have anaplasia and bizarre-shaped nuclei here. But when they are only in this area, and the basement membrane is intact, this is in situ, okay? So, at first we have in situ tumor. We call them carcinoma in situ. And if we find them in this stage, all of the tumor are curable. So understanding about this and finding the tumors in this stage is very important.
Second stage? Second stage?
In this area that they are in situ, I said that this stage is carcinoma in situ, which the basement membrane is intact, and we have no neovascularization. We have no neoangiogenesis in this stage. But after that, this tumor starts to movement. At first, they start to reach this area of basement membrane, and after that, they enter to the underlying stroma. So we have the invasive carcinoma. So we have two types and two stage in carcinoma. At first, they are in situ, and after invasion to the stroma and after neoangiogenesis, they are invasive. Okay? So, early in their growth, as I said,
Don't we have 4 stages?
4 stage.
Yeah.
What do you mean, 4 stage? Which stage? Can you name them?
The both, I don't remember the names.
Talking about the differentiation. He's talking about the differentiation.
Differentiation, you mean well differentiated tumor, moderately differentiated, and poor it's another thing. I want to speak about them in the other slides. Yes. So, after angiogenic switch, we have termination of this stage of quiescence of the tumoral cells. As I said before, everything in our body needs some pro-factors and some anti-factors, okay? As we said in our previous session, we have some pro-apoptotic factors and some anti-apoptotic factors, okay? And here we have some pro-angiogenic factors and some anti-angiogenic factors, and in the normal situation, in the normal people, these two groups of factors are in balance with each other. But in the tumoral environment, we have imbalance between them, and the tumoral cells need more pro-angiogenic factors, and they want to inhibit the anti-angiogenic factors. The molecular basis of the angiogenic switch involve increased production of angiogenic factor and loss or inhibition of anti-angiogenic factors. These factors may be produced by the tumoral cells and also by inflammatory cells. You know, the inflammatory cells in the environment of tumor is very important, and they can cause many change. They can help sometimes to the tumoral cells, especially the macrophages. The macrophages can produce some factors that are pro-angiogenic, and they can help to the tumor for making vessels and for giving nutrient and energy and oxygen. And also, the other cells that can produce the pro-angiogenic factors are the stromal cells in the tumor. Okay, in any tumor, we have some tumoral cells, for example, in this area after invasion to the stroma. Okay, after invasion, we have a mass-like proliferation of the tumoral cells here. For example, like this, we have invasion and very infiltrative pattern of the tumoral cells. For example, this is about squamous cell carcinoma. Okay, in each tumor we have the tumoral cells, and also we have some inflammatory cells in the background. This is the environment of tumor. Okay, for example, some lymphocytes, some T lymphocytes, also some macrophages. You know that macrophages have abundant cytoplasm like this, but these lymphocytes have little cytoplasm. And also we have some stromal cells in the background of the tumor. We have you know that the stromal cells are spindle-shaped cells, for example, like these elongated spindle-shaped cells in the background of the tumor. So this pro-angiogenic factor can be produced by tumor cells, background stromal cells, and also the inflammatory cells, especially the macrophages. Okay, about the enzyme proteases. We know that proteases is one of the most important enzymes which is produced by the tumoral cells and also by the inflammatory cells and the stromal cells. And these enzymes are involved in regulating the balance between angiogenic and anti-angiogenic factors. They are elaborated, as I said before, directly by tumoral cells or from stromal cells under stimulation of the tumoral cells. Many proteases can release pro-angiogenic growth factors that are stored in the extracellular matrix. Conversely, the angiogenesis inhibitor like angiostatin and endostatin, these two factors are anti-angiogenic factor, are produced by cleavage of plasminogen and collagen respectively. It means that by cleavage of plasminogen the cells can make angiostatin, and by cleavage of some specific type of collagen we have endostatin, and these are anti-angiogenic factors.
So what's the word of it? What's the function of it?
Affection of this, I said before that the pro-angiogenic factor and anti-angiogenic factor in normal situation are in balance, okay? So the normal situation need these two factors for stopping angiogenesis.
So body is degrade the collagen and basement membrane?
Yes, in normal situation we have many enzymes. There are many enzymes in the body that they can start, for example, in any injury. In normal situation, we have some types of injury. For example, in our skin bone. Okay, in this area, we normally need angiogenesis. We normally need making